Hello, everyone, and welcome to today's webinar, MultiFLEX Applications in Flow Cytometry. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter. To learn more, visit beckmancoulter.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Alfonso Blanco, Associate Professor and Director of Flow Cytometry, Conway Institute of Biomolecular and Biomedical Research, University College Dublin. Dr. Blanco, you may now begin your presentation.
Great, thank you very much. So, as I said, I'm Alfonso Blanco. I'm the Director of Flow here at UCD in Ireland, and that's why some people know me as Alfonso from Ireland. But reality is that I'm not Irish. So the first thing I will say is this is a disclaimer. So anything that I will say from here, it's my responsibility, and this is my view of cytometry and things that we consider as one way of doing it. But there are many other ways of doing things in flow cytometry. So as I said, I am not from Ireland. I'm from Spain. This is me a few years ago. I grew up a little bit. And I born in Seville, so the south of Spain, but really I'm from Galicia. All my family is practically from Galicia. And that's where I did my degree in biology, and afterwards I was doing my PhD, and it was working with this instrument here, a Coulter Epix Elite. That was a cell sorter, and that was working with plants. After the PhD, I decided to move in Ireland, and I was the first one running a core facility here in Ireland for flow, and that was a few years ago, back in 2004. And that was the first core facility of flow cytometry here in Ireland, and afterwards were quite a few other core facilities here. So the normal thing is growing up and expanding, and this is what the facility looked like a few years ago, back in 2004. I have a couple of analyzers here and a cell sorter at the end. And we were lucky to get quite a few fundings and grants, and we were increasing the amount of instrumentation that we have and also the variety based on the applications that were coming up, the growth of the technology, the needs of the users. And what you can see in my old lab is that we have practically everything fully full of instrumentation. And then we moved to a larger lab where you can see we have more space. That was in 2013. But the technology is moving and advancing, and several of the units that we have on that image have disappeared because they became obsolete or because they were no use anymore, and we have to replace it with new instrumentation. One of my main roles, and this is probably one of the things I enjoy more, is in training and education. Like I'm showing there, we are now running this summer school. We do lots of winter and summer schools, and this is the last one we are doing, the summer school that was on the 24th of June. What we do is a mix of lectures and hands-on, and we in the last year we have done a face-to-face together with virtual practical sessions. We have to readapt and grow and learn from the experience that we have, the bad experience in this case with the COVID, but it helps us to understand more things. So it's a combination of lectures and hands-on. So what we are trying to look is for instrumentation that is easy for the users to understand and follow up, that they are not scary. As I said, I'm involved not just in trainings at UCD. I'm participating in several different training courses and different workshops, like this one's organized by the ESCA, the European Society for Clinical Cytometry. Here with IFCC, we organize a couple of courses in Latin America, and that's where we are organizing many other actions as well, and schools. This one here was the first ISAC school at South America. I was in Brazil, in Sao Paulo, and I'm also involved with ExCyte. I'm the European distributor for ExCyte, and we organize several courses in different places. What is my main target as a core manager? My main target is basically introduce. As we are in an academia, we have to introduce students into the flow cytometry world. And we have to train them on the usage, but most important is to make sure that they understand the concept. Rather than training, let's say we are educating them into cytometry. Of course, as part of the role of the core facilities is help with the setup of each one of the samples and protocols, help with the analysis and reanalysis. And before all of that, we have to explore which ones are the options that they have, possibilities, design of the panels, etc. And in a day by day, when the users are running the samples, we have to fix some of the mechanical problems. It doesn't matter which one is your instrument, they will always have an issue, and we always have to fix it. Of course, one of the things we have to do is before we buy the instrument, we have to make some research and look at which ones are the offers that we have available on the market, what are the pros and the cons. And every single instrument, they have the big benefits and they have the not so good benefits. So pros and cons. It's like buying a car. When we are buying a car, we have our favorite one. Also, we look at the budget, but at the end of the day, we have to look is if it will be the perfect one for my applications, where I want to do what I want to do. And as I said, because I'm really involved in education on cytometry, and this is one of my passions, I want to make flow cytometry really attractive. By making it attractive and accessible to everyone, what we are doing is trying to attract more users into the facility, and this is something that we really need. It's a core facility. We have to charge for the usage, so more charges, more users, more charges. Then the administrator is happier, and that's at the end of the day is small business. But definitely, what we want to do is make solving problems. We want to make sure that everyone has their samples analyzed and their questions, the biological questions, are properly addressed and answered. So that's the philosophy of any core facility. And this is basically not just you as a user, that's me as well as a core manager. You're coming with the experiments that you want to develop, and sometimes we are absolutely lost. We cannot be the experts of everything. We need to learn. And the only way of learning is by asking questions and talking with the other experts and just joining actions and joining information to make sure that we are moving forward. So imagine that this is you nowadays, and you want to learn what's flow cytometry, and you want to know what you have to do to answer the question that your supervisor is telling you, how you do this, and you need to use flow cytometry. And you arrive to the lab and you see, okay, they have one equipment. And you see something like this, full of wires and boxes, and you don't know, and lots of screens. It's slightly scary, especially when you look at more details and you see all those wires started to be more and more scary, and suddenly you see the lasers there. Wow, lasers and fluidics and electronics and all those screens. And especially when you see all these danger signs around, you started to regret to be on the lab. And especially when you see all these biohazards all around the lab, definitely what you will do is basically run away from the lab. You don't want to be there anymore. So what we are doing, it's going to meetings and events like this one's organized by the International Society for Advancement of Cytometry, and it's kind of going to the supermarket. You have different offers from different technologies and different companies. And in 2008, there was one small, teeny tiny flow cytometer called Acuity from a company that was completely new, was Acuity in this case, and the instrument was a C6. It was a very different instrument because it was very small, very compact. There was no fluidics, big tanks of fluidics. It was kind of weird to see that kind of flow cytometer, and they were promising that was a flow cytometer. And also they have another staff on the electronics that was quite different, and was the way that the photomultipliers were located. They have 7 log decays. So all of that was kind of weird. But at the end of the day, it was a very easy to use flow cytometer. As you can see, any users were able to run their samples. This is one of the users that we have in the facility at the time. As you can see, he's a little bit upset, or he looks a little bit upset, but that's because I told him he has to wear lab coats and gloves, and he was not quite happy with that. But anyway, he was running happily this instrument and getting the data, which is at the end of the day what they wanted to do. As I said, it was kind of slightly different, and one of the big differences is that they were using peristaltic pumps to move the samples, and this is the filter that we have here, and these are the tubing moving everything into the flow cell. We have here two lasers, the blue laser, red laser. They were both collineal, forward scattered in the front, side scattered in a right angle, and then the photomultipliers were aligned in different angles than the right scatter. So it was kind of strange, but it was doing the job. And the other thing that was weird is to see that they have only one filter in front of the photomultipliers, where usually we are looking at multiple filters aligned one with the other. That was kind of different as well. And as I said, this logarithmic scale was great because practically everything was fitting. Nevertheless, not all the samples were properly set up or fitted into that logarithmic scale. This is a GFP. It's a very bright transfection that we have. And as you can see, the problem that we have is that it goes completely off scale. It was too bright. So there were two options. We can buy a neutral density filter, or we can use the filter on the next detector, on the FL2, which was a 58540, which is better for PE, not for FITC. But anyway, we could get the full distribution of the signal. Okay. So there were a lot of things that I really enjoy with this machine. Many things, and I really fall in love as soon as I see this instrument, the Accuracy 6, that later on has been bought by Becton Dickinson. But at the same time, there were things that I didn't like. I didn't like to lose one of the channels because my signal was too bright and I have to use it on that one. I like to readjust that situation, or the fact that we only have 4 detectors. Even if we have the blue and red with a flexible configuration, I like to have more lasers and more colors. And the reason is because the users were demanding more options. So usually love and hate, they're divided by a very thin line. So even if I love it, there were things I hated when I have to face the instrument because it was so easy to use, but at the same time, didn't have the complexity needed in a core facility. A few years later, in 2014, there was one instrument called Cytogene, was kind of different than the others, but was based on the Accuracy platform. And in this case, the instrument has 3 lasers, and you can analyze far more colors. It was 12 colors where you can analyze at the same time. And that was presented in Cyto, the ISAC meeting in 2013, sorry. And in 2014, show up as CytoFlex, and that's because Beckman Coulter acquired a Cytogene company. So in 2014, they showed the Cytogene that was shown in 2013 as the new CytoFlex platform. And one of the biggest advantages, rather than using photomultipliers, they're using a new generation, which is the avalanche photodiode system, so APDs, which are here. These are, as you can see, very small, and they were located in the back of all these mirrors that we have here. The light was redirected using this fiber optic, and one of the biggest advantage, and that's why they use it, is, as you can see on this paper published in 2008, and one of the authors is Paul Wallens, you can see that the advantage of the avalanche photodiodes versus the photomultiplier is that these ones are able to get far better signal, more resolution on the signal that you get into each one of them. More quantum efficiency means that things that they are dimmer becomes a bit more brighter, and that's a big change. We can suddenly detect signals that they are quite dim on the expression with no problem. Okay, and the other thing that we can see on this graph is that these are especially nice and very sensitive on the wavelengths on the red. That makes a big difference on the red emission when we are looking at APDs versus photomultipliers. The other thing that has an advantage on these instruments, Cytogene, CytoFlex nowadays, versus the VD Acquity, is the possibility of tunable dynamics. That means that basically we want to get this one like this in the proper filter. So we could modify, in this case, not the voltage. What they have is a modification of the gains, but at the end of the day, it's practically the same. And how this work. So this is how I see it. Imagine that you are taking this picture, okay? This is what you will have with a conventional flow cytometer. But if you increase this dynamic range, you can see that the picture is nicer. We can see more things in this space. And look at this cathedral. This is in St. Petersburg. You can see that cathedral there, but it looks like it's off screen. We would like to have that one in the center of the screen. So this dynamic range, with the possibility of being fine-tuning, what it allows me is basically to drop down the gains and put that signal exactly where we want it. So as I said, similar to the normal voltage. This is my signal, fluorescence intensity from the propylene iodide, very bright signal. As you can see, goes completely off scale. On this other detector, it's on scale. But if I want to place both of them in the same place, the only thing I have to do is decrease the gains that we are applying into the channel. And that's what we do. Similar, as I said, to how the photomultiplier works. With photomultipliers, you're changing the voltage. With these avalanche photodiodes, what you're changing is the gain. As I said, there was a change here. We have now the CytoFlex, and they have moved from CytoFlex, CytoFlex S, CytoFlex LX. This CytoFlex S has, instead of the 3 lasers that we have originally with the CytoFlex, has 4 lasers, and the CytoFlex LX has the possibility of getting up to 6 lasers with 21 colors, which means that we have a huge amount of flexibility. And now, in the last year, Beckman Coulter has released also something to complement this equipment, not just the possibility of looking at my cells with high sensitivity. They allow us as well to sort those cells. And this is a new member of the family, which is the CytoFlex SRT, the new sorter, using the same technology than the CytoFlex S, with the same configuration than the CytoFlex S, which makes a perfect complement to a core facility where you have the CytoFlex S to analyze your data, and then the users can move straight into the CytoFlex SRT and sort that population. So as you can see, with that combination, I'll be quite happy. In this case, this combination of CytoFlex S and CytoFlex SRT was during one of the training courses we have done. We were able to do a training of people analyzing and sorting at the same time. So this is the user, as I said, that's quite upset because it has some limitations, but now we put that big smiley face, not just because he's wearing the lab coat and I'm not upset anymore, it's because you can see you have far more capabilities now to analyze your samples, flexibility in terms of the excitation sources, flexibility as well in terms of the capabilities that you have for selecting the the wavelengths that you want. It also has the 96 volt play loader here and a very nice and easy stable fluidics platform. So this is my instrument, and now what is the next step? So the first thing I will do is check how sensitive is the instrument, which ones are the capabilities, and for that, usually we use beads. In this case, this was one of the first things I tested, checked the AP beads and the capabilities. And as you can see, in all the different channels, we can see how sensitive the instrument is. This is FITC, PERCP, and if you look at PERCP in the other system, you can see that the low end practically doesn't have any resolution. Even with the AP beads, we have the green laser, but also look at the capabilities on the APC 750. We can nicely resolve those populations. With the violets, same situation, you can resolve nicely the APEX from 450, 525. Violet 610, the low end started to be a bit weaker, okay? But the resolution that you have on the upper side is magnificent. So very sensitive instrument, very powerful for what we want to do, just basically working with cells. But usually people is thinking of flow cytometry, immunology, big cells, mammalian cells, and whole blood. But we also want to work with platelets, with bacteria, with viruses. Remember, we are in a core facility, so we have to be open to work with a huge variety of samples. We have to work with cells that are coming from tissue. We have people here working with retinas. We have people working in sperm. We have groups working in agriculture, in botany. So that means have to work with plants. We have to work with people working with algae, with phytoplankton. We are working a lot with groups here working with nanotechnology. And nowadays, we also have people working with the EVs and viruses. So therefore, sensitivity is definitely one of the issues, one of the main issues. So we need to optimize our instruments. It's not just as the instrument comes by default. We need to do something similar to what we do with the conventional flow cytometers, that they have photomultipliers, that you do the voltration. We do the optimization by gain tracing. Okay, we optimize the gains. This is a really nice video provided by one of the Beckman Coulter engineers, Hakim. And you can see we have here different tubes loaded with these gains at different settings, from 10 to 100 at different positions. And what we will see is those 8 peak beads and see how sensitive the instrument is at the different positions. So what we'll do is we'll put different channels and we'll keep one stable. In this case, it's fixing. So we have 1, 2, 3, 4, 5 peaks, and then the last peaks here, they're all together. In this case, in this channel, we have 1 and 2. They're quite broad, and look at all of these ones together in this other one. So what I will do is I will play, and this will be animation of how they're changing when we are increasing the gains. And what you will see is that these peaks, as soon as we started to increase the gains, they started to separate. Okay, you will see them in these ones at different points on the gain. So there we go. You can see how they're increasing. Now look at these ones. They're becoming very, very, very sharp. These ones are coming smaller. And one of the things you will find out is that this peak here suddenly arrived to the maximum position. It saturated the detector. And this next one will join. So that means that we are losing that capability of resolving very bright populations. So keep going. Let's go and play. And you can see this one suddenly is saturated. This one is saturated. Now we're expanding on the low end. We can see more separation, but suddenly starting to increase. Higher the gain as well, what we'll get is as well a multiplication on the background. And the populations on the low end are not quite well resolved. So what we need to do is try to arrive to a compromise. These are the different detectors, and these are the different samples that we analyze at those different gains. And what we can see is that there are some positions here that the detectors are fully saturated. On the other side, we can see some of the detectors, they do not saturate. We can see as well that some of the detectors, they get a huge amount of background. Some others, they are not affected. What we are looking at, we are looking for gains that don't saturate the detector, but also that we have not increase on the gains. What we are looking is for those gains that are right in the middle, those ones that are giving us the maximum distance between your negative populations, your dims, and your brights. For that, rather than using the eyes, we can look at the statistics, and we'll try to identify them by using staining index, signal to noise, robust coefficient of variation, and we'll try to identify which one is the position for that gain. A gain that is giving us confidence that we'll get a good signal. And we'll do it for each one of the channels that we have on the instrument. That way, we'll get a minimum, a position for starting, and that we have the confidence that it's quite close to what we want to use for most of our samples. Right, so we have an instrument that is quite sensitive. What we can do, as I said, we are in a course facility, so the applications, there could be anything that you can imagine. Any applications that you have in mind, it's coming through the door, and we have to face them. And the nice thing is, for most of these applications, we always have a solution with the MultiFLEX platform. And remember, not just for the analysis, we can also start to sort with these instruments. So we have the MultiFLEX SRT. So the first thing we will consider is, what about the autofluorescence? Are your cells autofluorescent? What is the minimum autofluorescence? Remember that all of you are autofluorescent. What is the autofluorescence? We put a source of light, and suddenly your characteristics on your cells are transferring, changing the wavelength, producing a different wavelength. So in this case, we are collecting this white light, and we transfer this into a red light. Older you are, more autofluorescence you will be. And there are many things affecting this autofluorescence. For instance, the size. Not just the size, also the composition, the content of these cells, how many vesicles you have. That will change as well that autofluorescence level. And these are things that we need to consider when we are doing any kind of analysis. For instance, here we have one sample where we have the cells that we expose to nanoparticles. These nanoparticles are uptake, and that produces a small change in my forward scatter. As you can see, there's a small increase from sample control to a sample treated. Changes in autofluorescence will be also reflected on my side scatter. This one looks like far more complex than this one. So one of the things we cannot forget is that these changes on this formation of my cells will affect as well my side scatter. Side scatter is a good way of looking at physiological morphological changes on my cell. And you can see there, it's clearly showing up that increase on my side scatter when we have these events inside my cells. As I said, autofluorescence is there, especially nice to consider when we are working with plants, and sometimes is a really dramatic change. And this is a problem when we are trying to put some markers. Like, for instance, if we are trying to use propionate light to label the DNA content of these plants, and we want to work with those plants and know how much DNA they have, the problem if we use propionate light is that we'll be overlapping on the emission with the chlorophyll. So we need to consider that. But not just the chlorophyll. If you think on these cells, they have many other things. They have cell walls, so they will have lignins. They will have beta carotenoids. They will have polyacetoates. They will have a lot of different things in their composition that will affect the emission of that autofluorescence. So these are things that we need to consider. And this is a clear example. These are the plants I was doing my PhD with. These are ferns. And when we look at the structures that we have on the back, which is basically the spores and the organelles that are producing the spores, the sporangia, we can see that depending on the wavelength that we are using to excite and collect the signal, we can see, for instance, the spores here, they're practically not producing any kind of emission. Meanwhile, these rings here that we have these cells of the ring, they're producing a huge amount of fluorescence in this wavelength. But if we change wavelengths and we move into this other one, the spores are the ones that are producing the highest amount of autofluorescence. And the ring, the cells of the ring, are practically no fluorescence. Okay? So depending on the structure, depending on the cell that you're using, there could be more or less fluorescent. And this is something that you need to consider when you're working with your samples. So this is, for instance, one example of one of the plants. We make a chopping, we analyze them, and we can see exactly that effect. We have some of these channels massively affected with those autofluorescence, and other channels that they have practically no autofluorescence, like these ones. So if I have to select one marker to distinguish or differentiate anything, those will be the ones I will use because that will be easier to recognize a big change rather than these ones that they are practically saturated because of that autofluorescence. Is that a problem? No, usually it's an advantage. And this is something that we have to start to learn, use the autofluorescence as an advantage. In this case, my users are trying to compare the production of a protein that is when we have these two cell lines here. And as you can see, both of them, they have similar forward and side scatter. So if we put them, both of them growing at the same time, using forward scatter and side scatter will make quite different. We can use side scatter versus PE here. Okay, you can see side scatter is practically the same. PE's signal that we have there is very similar. So when we have, in this case, the production of The protein that we are interested in is this one here, okay? And what about the other expression is here? What we cannot differentiate is the production of this one and the production of the second cell line when they are coexisting, when we put them both to grow at the same time. So rather than using cytoscatter, what if we use another detectors? Remember, we have a lot of different channels there, so we can use them to characterize the autofluorescence. So you can see, in this case, FITC channel is higher on this cell line than this other cell line. And when we run them both together, difficult to differentiate which one is the protein produced by one cell line, which one is production from the second cell line. If we change now to APC, now the other cell line is brighter on that channel. The autofluorescence is higher. But if we look at Pacific Blue, coming from the violet laser Pacific Blue channel, the 450, we can see how different those two cell lines are in their minimal autofluorescence. When we analyze the production of one, doesn't overlap with the second one. And when we look at the production of the protein, we can see that it doesn't overlap with the other. Even more, what we can see is that when they are producing the protein, the level of autofluorescence of this cell line drops down. And this hasn't been seen if we haven't played with these levels of autofluorescence. Okay? So look at other channels, not like just to put colors, also to understand how is your cell working. This is another example where we are looking at some particular detection of receptors on the surface of my cells. And you can see the blue line is my control. And you can see that the blue line is giving me 2 peaks. The autofluorescence is not equal. Your cells are growing, your cells are expanding, your cells are proliferating, your cells are doing things. So the autofluorescent level will not be the same. It's heterogeneous. Anyway, we have the marker, and we can see a clear increase on my detector using this laser and this bandpass. What if you use a different laser, exactly the same bandpass? It's an optimal laser, and this is the advantage of having the CytoFLEX. We can select different wavelengths for excitation, and we can use the same bandpass filters. And you can see by using the optimal excitation source, we can get a better resolution of my sample. If we try with the different filters, we can see we can resolve as well those dim, medium, or bright signalings. But if we use the perfect combination of lasers and detectors or filters, we can clearly distinguish a very positive signal. And this is what I like on these instruments, is the flexibility to choose excitation sources and emission, and playing with these ones to try to optimize and maximize the information we get from every single sample that you have. In this case, we are trying to work with samples that they are trying to do an uptake, and the group, in this case, is from Professor Kenneth Dawson. They wanted to look at the receptors that we have and see if there is any relationship between the receptors and the nanoparticle uptake. So we have here nanoparticle uptake and the marker to detect that receptor. And you can see, as I showed you before, we have the negative, and then we have the 3 different levels of emission. And now we put the nanoparticles. This is my control without the marker for the receptor. What if we put the nanoparticles and the receptor? We can see that they are exactly in the right place where we expected to see. So resolution in this instrument on the CytoFLEX is fantastic. Let's go and talk now about viability, because it's definitely something that we need to consider. Even if you're not measuring alive cells and dead cells, we need to look at viability as a marker to remove dead cells from our analysis. Excuse me. Right. Usually, if we are working with cells in culture, what we will do to look at cells, we just select forward and side scatter. We can see here the junk, and we put the polygon to try to remove the junk. And we can see here that we are trying to do the singlets. So we'll use forward scatter area, forward scatter height. Here we can see my cells, and here the aggregates. So we'll put that singlet gate there, right? Now, this is the same, but now we have my samples treated. and they are dying. So we can see here my dead cells. Let's put the gates exactly in the same places. But is better this one or this one? There's people that prefer this one. There are others that prefer to use this rectangle. Singlets, we'll select that one. Fine. And now it's more difficult to select where is the junk and where is my dead population. We cannot see the clear distinction between them. And same here. We're looking for singlets. What is this population that's pulling out here? I have no clue. Let's change the scale from linear scale to logarithmic scale. And in the CytoFLEX, you can change as long as you want. In other instruments, you have to click and say, I want to record this in linear or I want to record this in log. The CytoFLEX has the possibility to select both at the same time and transfer them from one to the other. And I personally prefer to work in logarithmic scale, compress the data, and we have a better view of what's going on, even with cells in culture. When we look at these ones, we can nicely see my alive cells here, my dead cells here, and the junk, and I can clearly distinguish where the junk is. Even when we're looking at forward scatter area height, I can see my singlets here, the aggregates here, and what we have here is my dead cells. And this straight line that is going kind of a diagonal are my junk. So this is the way I will select if I want to do forward and side scatter. And this is the way I will look at them over here. You can see I'm selecting cells and I'm removing the junk, which remains in black. If we do the other way around and we select the singlets by selecting my dead population and my single cells and excluding the aggregates and the junk, and I go back to the sample, this is how it looks like. That gating here that we use here looks like it's practically collecting every single event that we have in alive and death. And that's what I really like to start with in my analysis. Let's have a look at how this could change from linear to logarithmic when we are having samples that they are happy and alive or death. You can see my alive cells are here, my alive cells here. We don't have the group of dead cells as we mentioned before there. How these ones are starting to be damaged and changing the position, we have more dead cells here and the aggregates on this direction. There we go. Now we put the gates and we can see cells that are alive, slightly damaged, and death. We can see here more dead cells, but they are already moving into the junk. So they're moving from here to there. And these are completely junk fragments already. So if this is the junk, as we can see there, these other ones that are now in black are the remaining. And you can see this is where we have our cells. So that's why I like to use that kind of gating to look at my cells. If I'm trying to analyze alive and death, that's the way I will select them, or just basically remove my dead cells from the analysis. When we are looking at samples, we have to be careful with the sample preparation. We have to be gentle and count the cells. And we usually have as well counting cells beads here. So we have accutates 3 minutes, 5 minutes, or 10 minutes. And here we can see more or less we have more cells here, but more or less is not good. We need numbers. We will compare this sample preparation with triple express at 3 minutes, 5 minutes, or 10 minutes. We can see that looks like triple express is better. But as I said, looks like doesn't mean it is. We need to look at the numbers. We need to look at percentage, 12%, 25%, 46. Clearly this one has more, 20, 52, 43. So it looks like this one has higher percentage. So we compare those ones, look like triple express has percentage-wise more, but doesn't mean that we'll have more cells. Percentage could be slightly different. We have to look as well at events per microliter. And this is the advantage as well on the CytoFLEX. It allows us to measure the true volumetric counting. So we can look at the number of events that we have per microliter. And definitely in this case, we have 534 versus the 422. So we have more cells per microliter than when we use accutates for 10 minutes. So this one is an example where we have used this method as well to optimize how we get more cells. In this case, trying to optimize this system, which is in this case for colitis, using a new device. Because when we are using the traditional methods, the problem is if we extended too much the digestion to get the samples that we want, we have a lack of viability. As you can see here, using traditional methods, only not even 30% of our cells are alive. And the other problem is when we are using traditional methods, like chopping, the chopping is not equal, it's not homogeneous. One day we are chopping more, some other days we are chopping less, depending as well on the blade. And that's when we were partnering in this case with Syntec to try to evaluate how is the MediMachine 2 working in terms of homogenizing the system to get always a repetitive way of collecting my cells, that there is no variation because I'm in better mood or bad mood. So we evaluated the MediMachine and different medical systems for this kind of tissue. And this is a work with Ashish Singh and Ulla Naus from my university. And as you can see, there's a clear improvement with more than 84% of the cells alive. And the best thing is quite consistent. It's quite reproducible over time. Also, we could see that there is no damage on the expression markers. We have tried in this case with different kind of cells. And we have seen as well with this one, so you can see it's mesenteric lymph nodes, Freyer's patches, and spleen, that the viability is always above 80%. So definitely working with these systems help us a lot on these analysis. Okay? We can get more cells and in better shape. And the advantage is not just the percentage is higher, it's also the number of cells that we collect back is much higher than before. So higher number of cells viable, and the most important thing, higher consistency. When we're looking at cell cycle, which is another key application, especially in this lab, and this is funny because every time I was looking at these waves on the COVID, I was looking at them as cell cycle. It's professional deformation, I think. One of the key things here is these detectors. We want to resolve those populations. We want to resolve cells in G1, S-phase, G2M. So getting systems like this one with that sensitivity will help us a lot. And I was really impressed with the sensitivity of the CytoFLEX because usually Propioners, right, will use it with the blue or the yellow-green laser and the 585, 610 filter. But this fluorescent is going not so far, really far away. It goes from PE detector or ECD detector on the yellow-green, PSI5, you can see perfect cell cycle. On the blue laser, we don't have a good cell cycle on FITSI, but we have it really good on PERCP. We have it as well on the red laser with APC and APC 750, and the violet laser, we have it on the red channels. And it's like, whoa, hold on a second, yellow and blue, violet, I could understand it, but red, let's see how is the excitation emission. If you see here, we have 561. We can excite PI optimally. 488, we get signal. Violet, not so much, but we have a lot of copies. And if we consider the red is not having the Stokes shift. Well, we are getting only this proportion here. It's very low, but as I said, with the APDs and the content efficiency, we have a lot of copies to even have a perfect cell cycle. And again, we can combine just cell cycle with other characteristics, changes like in the morphology, to characterize if my cells are growing and making it larger or not. The other thing we can look is for BDSAs, similar to ELISA, where we'll have the reports. And for that, we have usually a Luminex platform here, which is kind of automated to look at picograms per ml. As I said before, we have these kits and we can analyze them, fantastic, but sometimes we have warnings about the number of events that we are collecting. And when these instruments are producing this information, usually the company will say, look, it's your problem. You haven't mixed up my cells properly, etc. We get the statistics and we look at number of events we have in each one of the wells analyzed. What we have done is analyze the same plates parallel the instrument, the Luminex versus my CytoFLEX. We can get exactly the same positions that we have on the Luminex with really good separation resolution. And we also have the possibility to look at the number of events per microliter. Not just the number of events that we have counted, also the concentration per microliter. And we can see in our kits was quite inconsistent in terms of the numbers, and it was not just one plate, it was in several plates. And with this information, we could come back to the company. If we think now a little bit smaller and we go to microbiology, we have yeast and bacteria, etc., etc. There are several groups that are trying to do that. For instance, people that's working in genomics, and they use yeast as one of the um, uh, models, the live models to work with. In this case, they're working with yeast that they transfect with GFP and tomato or in cherry, and you can see the big differences between one and the others. Even we can see populations in the middle with different levels of this transfection. What they want to do, they want to sort these populations and not one, several ones. So it means I have to be here for many, many hours working with them side by side. Nevertheless, the new SRT is so simple to use. It's as complex as the fax area, but it's so simple to use that the instrument can be used by the user and they can spend as much time as they want during the weekends or during nighttime. The other thing that we mentioned before is that you have the 96 plate loader, and the idea is to work with plates. And we have all these bacteria analyzing. In this case, this is my scatter. We can see if my cells are alive or dead and look at the receptor and see if my bacteria, they have the receptor. As you can see, it's very important to look at viability because look like they have the receptor, but these ones are dead. Okay? It's better this position where they have the receptor and most of my cells are alive. They have really good softwares to show you not just if you have them or not analyzed, but also to look at the levels of viability or the receptor expression. So really nice software, not just for the acquisition, but also for the reanalysis. And these are different bacteria that different users want to use. In this case, they're producing some proteins that they, as you can see, they crystallize in red. So forward scatter, side scatter, we have the protein here of expression, but when we have Phe addition, you can see we have more red. And what happened is look like we have an increase on my side scatter. Remember, if we look at side scatter, this is correlated to these crystals, but also look at the fluorescence, clearly has increased the fluorescence level. Fantastic. Let's go to the brightest one because they will be the ones they have more protein expressed. So protein expression high, side scatter high. This is my target then. This is the one we want to sort. So what we will do is we'll look at this one again. This is my Cyto9. These are my bacteria, Cyto9, and we have a viability marker, in this case, DRAG7. When we have the protein expression and on this one, we will look that side scatter here is slightly different. When we look at DRAG7, we can see that there are a lot of dead cells here. And what is happening here? It increases here because of the DRAG7 overlapping, but at the same time, we can see that most of them are dead. When we look only at the live cells, the level of expression is very low. But now we have this group here that we know that they're bacteria, that they are alive, and they have the protein of my interest. Those are the ones that we want to sort, not the other ones. Okay. How we sort? We can sort in agar plates, which is really nice. We can see here, hold in for a second, plate again. So you can see that in every single movement, we are putting one single bacteria. And afterwards, we just grow them. Okay. So here we can see that each one of those colonies are coming from my bacteria, except the first one, this middle one that we have 10 bacteria, and that's why the colony is slightly bigger. We have done exactly the same thing with the SRT in collaboration with a group in Poland called SAMPROBI, with Joanna and Kasia and the help of Michał Miał. And we were preparing the samples and selecting again those bacteria we want to check. We want to check if these are my bacteria alive, these ones are dead, and these ones we don't know if they're alive or dead. So we just selected them and we place them into an agar plate and we sort them. And you can see these are my alive, and these ones here are the population that we have here. They haven't grown in that agar, but they have tried different buffers and different medias, and in different conditions, they were able to get those cells that look like damage because they are in between one and the other, but in reality, they could recover them and they were alive. We can select, if we have multiple different kinds of bacteria, we can select them, put them in different colonies, and later on do a PCR to know which one is which, identify the different subpopulations. Let's go now for MV characterization, very small particle detection. For that, we need a very sensitive instrument. Why the MVs? Microvesicles, extracellular vesicles, exosome, they are all found in different body fluidics. They can be found in interspatial space, and usually they are coming with the antigen content from the cells that they are coming from. They usually have membrane bounds with vesicles that they contain phospholipids, and a few years ago they were considered as cellular debris, and that's why we didn't pay attention. They were the junk that we excluded in our analysis. But what we have found, what people is finding, is that those vesicles that contain mRNA and proteins, that they can be transferred and transported between different cells in different parts of the body. Some of the others, they can be used to remove misfolding proteins, cytotoxic agents, metabolic waste. So when I was talking with people and they were explaining me this, that they could have different sizes, different compositions, the content inside was there, and they want to classify and characterize, what came to my mind is basically this: garbage. We have different kind of garbage with different kind of containers that they could have different things inside. And as our famous Howard Shapiro used to say, garbage in, garbage out. So why do you want to do this? Oh, we want to classify them. Okay. How do you classify them? If we think on the kind of garbage that we have, we can think on this kind of bags, but we have these other kind of bags. So the characteristic and where it's coming from is clearly different, and what it contains is different. If we look at this bag, it's clearly different. And again, cargo and content could be quite different. But they will help us to characterize what is what and what they contain. And this is what the users want to do, characterize these different bags. But when we look at each one of them, we can see the refraction index is different based on two things: what they have outside and what they have inside, the characteristics of the package and the content. And they wanted to do it by flow cytometry, but flow cytometry is designed for cells, mammalian cells. So this is stolen from Edwin Valdepore's presentation, and I really like it. This is how a cell will look like in a flow cytometer. And this is how an EV, an extracellular vesicle, will look like in a flow cytometer. Quite small. So what we need is a very precise instrument to analyze. So what it has been found is that the ultraviolet side, the violet side scatter is more precise for very small particles. And the CytoFlex LX has that option to detect nanoparticle resolution. So this is a technical note from Beckman Coulter. You can see here, violet side scatter is an optional configuration for nanoparticle resolution. To be able to separate clearly the noise from the different kinds of, in this case, sizing beads. Okay? These are the dragon green beads analyzed on the CytoFlex by John Tiggs and Basilis. So what you do, basically on the instrument is coming with this violet filter here in a detector that is blocked. And what you need to do is just basically change those two filters from the position. So you remove the 405 and you place it into 450 just by pulling it out and plugging the other one in. And you have this one. What if you have to combine them with different markers? For instance, there are a lot of people working in EV that they want to use the brilliant violet 421, which will be analyzed in this one. So what we'll do is move this filter, the 450, to the next one. We'll place the 405 here, and this 525 will occupy that position. Is this new? No, it is not new. We have this violet configuration, or the usage of violet side scatter, in instruments like the Attune NXT. And they use it in this case to recognize the erythrocytes and exclude the erythrocytes from platelets and leukocytes in the no wash, no lyse system. So you can see here the erythrocytes, and you can see here the platelets and leukocytes. These ones, they have a different way of scattering the light more into the blue laser than on the 405 laser side scatter. The other thing before I move on into how we use it on the EV analysis, the other thing we need to consider when we are working with EVs is that this one could be coming with many others. And when we are looking at this big amount of EVs, they will be looking like a summary of all of them together. So that means the signal will be much larger and the signal will be much brighter than when we are looking at each one independently. And this is what is called the swarming effect. What we need to do when we are working with EVs, very small particles, is make a series of dilutions to make sure that we are working with one single event at a time. And there are two things on the CytoFlex that is telling us, be careful with that. The first thing is the increase on these scatter values on the median fluorescence intensity. And the second thing that we can see is the number of aborts is quite high. When we are doing dilutions or a series of dilutions, we can see the median drops down, the groups are much tighter, and the number of aborts decrease much, really, really dramatically. So now we can know that we are working with single EVs versus this one, which is a swarming effect. The other thing, if we want to make sure that we are working with single events, even if we have a very low number of events, is trying to remove the aggregates. Again, area versus height. We use in this case the violet side scatter. It's quite difficult to see where is the border between aggregates and singlets. What I found is by using the height versus the width here, we can nicely separate my single events versus the aggregates. I will use this one, and now I apply this gate here, and you can see how tight is my resolution, which is far better than this gating here. So I prefer to use violet side scatter height, violet side scatter width to differentiate my single population from my aggregated particles. And the other thing I want to point out is that apart from the violet side scatter here, we have another scatter here, which is coming from the red side scatter. So what we can use is this red side scatter and transfer into any of these detectors here, and then we'll get the red side scatter. Red side scatter, what's that for? Is it an advantage or disadvantage? You will find out now. And I find that it's a very good and advantage position. So these are the mega mix that everyone is using or some groups are using to try to set up the instruments for EVs. FITC versus side scatter blue with the localized might beads. Here is forward scatter, not good resolution for small particle detection. And this is how my particles look like, my EVs will look like with that particular setting. When we look blue versus red, you can see it's following this diagonal, and now we have the particles here. So it's a big advantage to have this one. So these are NIST, a series of beads that they have different sizes, 80, 100, 150, and 300. And using this case, the violet side scatter. Uses forward scatter, blue side scatter. You can see only able to resolve the two larger populations. This is the violet. We can resolve the 4 populations, and this is the red. In this case, the red is able to resolve the 2 top populations, but with more sensitivity than the blue. This is how it looks using now the Rosetta Beads from Exometry. You can see blue versus red, and we can see the 3 different side scatters, and they are really good for resolving these beads. But if we look at red versus violet, the red has better resolution than the violet side scatter on the low end, but it's better on the higher end compared to the red on the violet. When we look at the Rosetta Beads, this is how it looks on the 3 different combinations of blue, violet, blue, red, red, violet, and you can see that they are following more or less more red than violet. When we look at the megamix, again, we can see that they are more red than violet, and when we look at the NIST, we can see that in this case, the NIST is more violet than blue. So the combination of these 3 different scatters will help us to understand and find out those different subpopulations in a better way than if we use them individualized. When we look at the apogees, which are a different set of beads, and some of them they are green labeling, we can see the advantage of working with the red and violet or red and blue is that we can differentiate that they are kind of 2 different diagonals. When we look at this one here, it goes to the top side on the blue versus red. Those are beads that they do not have the green color. And when we look at the top side, you can see here they are more red than blue or more violet than blue. Those are beads that they have the coloring on the green. It makes sense because as soon as we have these marbles and we color them, the refraction index change. And because the refraction index change, this relationship of the different scatters on different wavelengths can be affected as well. When we look at these nano in different kind of analysis, in this case, they are nanoparticles, and we look at the blue versus violet, looks like clearly violet is more sensitive to resolve them. When we look at the blue versus the red, it's basically the same. It's more red than violet. But when we look at the red versus violet, they follow the diagonal, and this is helping me to differentiate the nanoparticles from the noise. Interesting enough is that when we have the red side scatter and we put these nanoparticles with protein corona, the protein corona is giving us a drop down on the side scatter. Again, similar effect to what we have seen before with these nanoparticles that they have the FITC color around them. So I was thinking, probably it's me, probably it's my system. So I talked to my colleague and friend, John Teagues, and I said, look, could you repeat this assay with your instrument? So we have the same beads. This is how they look like on my instruments in Dublin, and this is how they look in Boston. As you can see, the same patterns are happening. Now we combine the NIST and the apogee, and we want to see if we are having a diagonal or the same relationship between the different beads, or they are randomly placed and located. As you can see, depending on the composition or depending on the structures, you can see that they're not following a diagonal. They're jumping up and down in these diagonals. So keep talking about these particles. We were working with a group of researchers that they wanted to compare two different insecticides that they are coming into these kind of balls, introduced into them. These balls, they attach to the skin of the insects, and then they release the poison. Well, the size is really variable, extremely variable. And the problem is that they break down and they release the poison before they arrive to the animal. So the effectiveness of the treatment drops down. So they wanted to characterize the quality of these compounds. If you use microscopy, it's very difficult to characterize them. Even electron microscopy will not be able to do it. So we decided to go for flow cytometry. So this is how they look. And we use, again, violet side scatter, blue side scatter, and red side scatter. As you can see, the profiles for each one of these beads is clearly different. Not so much when we look at the fluorescence, except in a couple of channels where we can see slightly different. So we combine this with ratios, and we can characterize the ratios and the different scatters, and we can see the big difference on the patterns between fresh, in this case karate, and dry karate, or fresh lambda, which is the other one. This is lambda fresh, and lambda dry. So to characterize them, we put the statistics in the middle, and we can see the differences between fresh and dry, and the difference between each one of those ones, and we can characterize the different insecticides. If we are looking at, these are my megamix, and now we want to look at EVs. EVs coming from cells in culture. We can see here that the combination of violet versus red is providing us far more resolution to resolve, in this case, 1 to 3 populations versus just one, if we use blue and violet. This is sample 1, and it's able to resolve 1, 2, 3, 4 versus 1 again on violet versus blue. Here we can see 2, here we can see 1 and 2, but also we can see this tail here. This tail is coming in some cases, not in all the cases. This tail is coming when we have the most of the events coming from this side. You can see here too, we have the top and the bottom. Especially we have the bottom, we can see this tail coming from that one. Okay. So again, this violet-red combination is allowing me to characterize things that with just the combination of the violet and the blue, we will not be able to differentiate. What about the EVs coming from samples from plasma EVs, for instance? So we have an XM5 and CD markers, and the combination with red and violet also will help us to characterize them. So look at this one. This is samples from the same patient on day 1 and day 3. We have this population here that doesn't appear on day 3, and this one that shows up on day 3 and is not coming in day 1. When we're looking at these rare events, that's fine, but when we are looking at these day 1 and 2, we can see 2 populations with 2 different levels of CD expression. There's nothing in the middle between those 2. When we look at this population here, we can see that there's only one group of events. And interesting enough, they have intermediate expression between those 2. Look at those 2, and the next one is in the middle. This one, 1 and 2, bright and dim, and this one is intermediate in both of them. So it means that there could be different levels of expression. If we increase, in this case, the size of my EVs or the position of my EVs on the scatter, we can see they have larger expression of those markers, bigger EVs, more markers associated to them. But if we go to this parallel population here that shows up on day 3 and disappear on day 1, there is no expression of the marker. So it means that the origin of those EVs that they are changing in their scatter is clearly different between the position on more red and less violet. We could think that this is an artifact that could be an issue with one of the samples. We repeat in different patients and different days, and we can see they follow more or less the same pattern. We look at the statistics, and this allows us to identify different biomarkers for different patients. Okay, we can identify, can we sort? And this is coming from John Tiggs again. This is a SRT, and how the SRT is able to resolve these NIST particles at different points here. And you can see how small we can go with these NIST beads. So you can nicely see from 80 and 90, we can nicely resolve those populations. What about EVs? In this case, these are background, and what he has done from the sample was trying to sort those EVs that they're, in this case, they are GFP virus-like particles. So he was sorting these particles with no issues. And if you remember, I was mentioning about these no lyse, no wash. How does it look on the CytoFLEX? Practically the same. It doesn't look the same, but if you see here, these are my erythrocytes. This is blue versus red, and this one here is red versus violet. When we look at the leukocytes, they're coming exactly in the same place that we have them on the Attune NXT, with the advantage that we can see also with the red combination. Let's go closer, and we'll go now to the platelet section. And what we can see is in this platelet section, we have different shapes of these platelets, which means that we might be ending up with new subpopulations of platelets. We don't know. The next step is trying to sort them out. And Jordi Patrice just challenged me to try to identify if these different kind of neutrophils, they could be also differentiated by these different scatters. So I look at them, focus into those populations, and try to see them with more resolution into these violets. You can see in this diagonal here, I track here, that these events are always in a different position. When we use the multi-radial, you can see that we have a cloud of events floating around the main populations. We have used the same concept in this case for phytoplankton, and we have characterized different kind of phytoplankton, not just on the chlorophyll and all the autofluorescence, also using the violet and red side scatter. And what we have seen in these different cultures with more detail is that by using violet and red combination, we can identify multiple populations, which is clearly related to the different structures and the different compositions and the different ways that these structures that we have on the plankton. So we help, with the help of these scatter characteristics and the fluorescence, we will be able to identify with more precision the different subpopulations. And during this workshop that we have, the symposium organized online, there was one time in here that I had to do a practical session with EVs. My samples were wrong, and suddenly I identify a bottle of tea over there. I say, okay, they have bacteria, they have yeast, they have lots of things. Let me see how these look like in my flow cytometer. So this is the setup that we have. This is the tea, a sample of the tea collected and analyzed using the violet and red. And I can see this population here, probably the bacteria, and something else here going outside of the scale. So I got one sample from yeast that they were transfected, the same ones I mentioned before with transfection with GFP and M-cherry. And here we can see the GFPs and the M-cherry on the yeast. I tried to localize them. This is off the scale. I just need to drop down a little bit again, put them on the scale, and see where the yeast are falling. And now I analyze again the previous sample of T. And when we compare closely now, where is my population of yeast, and I put a gate around them, and I look at these ones here, the yeasts are falling here, and these ones here, then they're bacteria. In a different angle, with a different relationship between the violet and the red side scatter. When we look at this with more detail, we can see not just that, also a population right in the middle, different than this other population. So what we will do next, if we want to identify, as we have done with some probe, we just basically sort them, put them onto agar plates, grow them, put them on the PCR, and identify who is who. First, using those characteristics of a scatter, but don't forget about the autofluorescence. They also have differences on the autofluorescence. So we'll use autofluorescence, scatter, sort them, and then characterize them with proteomics, genomics, or whatever you want. If you want to know more about the different kinds of ways to analyze EVs, there's a series of webinars that John Tiggs and I have created during the pandemic. We were bored, so we created 5 different videos, also with the support from Beckman Coulter and all the support to do this. But with this, I want to just thank a big support I received from Beckman Coulter during the last few years. Also, LabPlan, the distributor of Beckman Coulter here in Ireland. My colleagues and friends from San Provi, Beth Israel, John Tiggs, and Brandy, San Provi, Joanna, and Kasia, and University of York, Karen and Peter. They were fantastic and they helped me a lot. Of course, Tim Bushnell in ExCyte. And bigger acknowledge to all the users on the facility that they are accompanying me during this process, and especially to these 2 users, Javi Blanco and Dani Blanco, my wife, and the core manager, of course. You always need to acknowledge the core facility manager. And with this, I'll stop my presentation and open to any questions. Thank you.
Thank you, Dr. Blanco, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is it possible to perform kinetics with the MultiFLEX?
Well, thank you for the question. In fact, kinetics is one of my favorite topics. That's when I was working with the Acri. If you remember, I mentioned that we have peristaltic pumps. MultiFLEX have also the peristaltic pumps, and it allows us to do continuous kinetic. Means that when you have a pressurized system, you have to stop, make a gap, and then you cannot see the events for a while. Put your drugs or any kind of stimuli that you want and just upload it, and then you see the kinetics. In that process, you can lose a good amount of information that is not there. But because we have these peristaltic pumps, we can continuously analyze the sample. How to do it? If you look at the MultiFLEX, we can just remove the lateral side. That will give us a bit more space, and instead of using the normal FACS 5 ml tubes, you use ependodes. On those ependodes, you can load from the lateral side your stimuli, your drugs. Meanwhile, the sample is running. And that means that we are speeding up the time to get your sample through the flow cell. And usually, if you run it around medium flow rate, it will take around 3-4 seconds to start to see the cells when they have been interacting with your drug or your stimuli. So MultiFLEX is a really good platform to do any kind of kinetics in real time.
Great, thank you. And it looks like we have time for one more question.
Go for it.
We have a MultiFLEX, and we are using the default settings that are coming with the instrument. Is it good enough, or do you recommend doing the gain tration?
Yeah, so thank you for the question again. And this is something that came up in many, many different training courses, and many of my users are asking, this is the default. So no, the default that is coming from the Cytometry is telling you basically the instrument is performing well, and it will give you the data good enough. But if you perform the gain tration, as I mentioned in the talk, you will see that your signals will massively improve. Especially on the dim side, you will be able to characterize better your weak populations if you perform gain tration. It's a very simple to say. You will perform in one day, you save this new settings as your default settings, your new default settings, and once you have it, don't modify. You can optimize it later on for each one of your experiments, but that will be a good starting point where you will have maximum sensitivity on the instrument.
All right. Thank you, Dr. Blanco. Do you have any final comments for our audience?
Yeah, I usually recommend the users to try the instruments with their own samples, and this is what I will tell them. And if you have the chance to work with the Cytoflex, you will be impressed by two things: the flexibility that you have, the sensitivity that you have, and how simple to use the software it is, but at the same time, how complex you could do your analysis. And this is what I wanted to show up during this presentation. You can do multiple things in a very simple way, and at the same time, enjoying your analysis.
Well, thank you again, Dr. Blanco, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Till next time, take care, everyone. Goodbye.